Hello everyone and welcome to Lab Manager's Tech Trends webinar series. My name is Trevor Henderson, Technology Editor for Lab Manager, and I'll be moderating today's discussion which will focus on automated liquid handling systems. Today we have a panel consisting of 4 experts in the field who will discuss a variety of technologies to transform your lab and reap the benefits of automated solutions. We like our webinars to be very interactive, so we encourage you to send us your questions at any point during the presentation, and the panelists will address these during the question and answer session to follow. To ask a question, simply type your query into the Q&A box, which is located at the bottom left-hand side of your screen. We'll try to address as many questions as possible during the question and answer session, and if we happen to run out of time, I will forward any unanswered questions to the panelists, and they can respond to you via email. Additional resources may be accessed by the resource window, and you may also move or resize any of the windows simply by grabbing them at the top or stretching them at the bottom right corner. At this time, you may need to move or minimize some of the open windows to see the live view. This webinar recording will be available early next week on Lab Manager's website, and at the end of this webinar, we'll share that link with you, as well as the contact information for all of our panelists. With that, let me introduce our first speaker. Kasia Proctor is the manager of the liquid handling automation marketing at Agyant. She has a Master's of Science in Biomedical Engineering from California State University and has been in the life science industry for over 10 years. Kasia joined Agyant in 2014, and prior to that, she has been working with plate readers in their various applications, including integration with automated equipment. Thank you for joining us today, Kasia.
Thank you. Thanks, Trevor. Thank you for the introduction. What I wanted to talk to you guys about is, what are the things to consider when you are looking at automating your liquid handling. So just kind of a quick overview. Key things to think about is why are you considering automating? Is it because you have an increased throughput needs, or are you looking for better data? And by that I mean better reproducibility. In the past, we've seen a lot of customers automate because of increased throughput. Nowadays, I'd say that has shifted over the last couple of years, and there's more and more automating for better reproducibility. You want to control your sample prep early so that you get better results from your experiments. So that will, then determine how much walk-away time are you really looking for, what equipment you'll be looking at, obviously, and it will also control, the budget will be affected by that decision as well. The second thing to consider are the applications that you're running today. What are you trying to automate? But also, what do you think might be doing in the future? After all, this is a piece of equipment and investment in your laboratory, and you want to make sure that you get best use of it, today and then in 2-3 years as well. So you should consider the hardware flexibility. What are the things that the instrument can do, as well as software flexibility? So how can you change your protocols? Ease of use is a big one, and as I mentioned, nowadays when people try to automate because of the better reproducibility, a lot of labs no longer have automation engineer, which we used to see a lot in the past. And so that means that a lot of scientists are the ones that are setting this up and using the system, which is great. But also that means that you need to be able to have a fairly simple user interface. And I'll show you some examples as we go through the slides today. And then finally, you want to get references of who else is using the system, with their feedback, and just get feel from your colleagues. This slide just talks about different markets, and there's a lot of people on a call today, and I don't have the background on each one of you in terms of which market are you in, but I just wanted to show that you can automate the liquid handling needs in various markets. Obviously the solution will depend on what is your application that you're using, or different applications. And at Agyant, we have different solutions depending on where you are, what are you doing. And we have different solutions for you, a prepackaged solution that you can purchase depending on your depending on the market, depending on your application. So in these different markets, we'll have different hardware that serves the market. What you will see in this slide, and it's kind of showing you a lot, so we have a lot of different solutions, but the key here is our Bravo platform. And you will see a Bravo several times in this slide, and that's because we use it for various applications. So you can have a standalone Bravo. You can have a Bravo that's configured for next-gen sequencing. And then we also have an assay map Bravo, which I'll talk about in more detail later. On the left, you will see the workstations. So what we're able to do, and this is really more if you're looking at automating because you want to have increased throughput, then you can build out your workstation and basically will allow you longer walkaway times, and it will allow you to process more plates. And then in addition, we have some equipment like the centrifuge or plate lock, which will help you process your plates. It doesn't necessarily do any liquid handling, but you will need it as you have your plates. So that will be part of your process. And then obviously there's software that comes with the consumables. Tips are a big part of the liquid handling. So all of that is we offer as Agyant, and once you buy a package from us, then we also guarantee the performance of the whole system. This next slide talks about, again, our Bravo system, which is the base for a lot of the configurations that I was talking about. Bravo is a nice and compact design. You can quickly change heads on it. We have some, I'm not going to show it here, but if you go to our website, you can see some nice animations of how the system works. And we have also various accessories that you can put on deck. One of the things, you can kind of see it in the image here, depends on your, I guess, screen size, but there is a gripper on deck. So you have 9 positions, and with the gripper, you're able to move the plates around, which really will allow you then to process more plates and increase your throughput here. There's also an option of having a through haul. Basically, if you want to put a truss under it, then you can get rid of the used up tips. So there's a lot of flexibility with this design. This shows you, when we talked about the hardware flexibility, we have 96 heads, we have 384 heads, but what you can see here is even when you have a full head installed, you are also able to use partial heads and use a couple rows, couple columns, or even a single tip. And so this really helps, especially if you are thinking of automating because you want to have better reproducibility of your samples, and you might not necessarily have a full plate to process. Well, you can still use Bravo to help you with your pipetting needs. I talked about, I mentioned the fact that you should consider the software and the flexibility of the software, and that ultimately means also the ease of use. So we, in the background, we have the VWorks software, which is industry recognized. It's been around for many years now, and a lot of the automation engineers, as I mentioned, those are our users in the past. They really liked the VWorks software. It gave them a lot of flexibility, but you really needed to understand how to process and how to write the protocols here. So you have that flexibility in the background should you want it. But now we also offer these prepackaged solutions. So the blue screen here is really your user interface. And what you can see is that you have it tells you what plates should be where on the deck. And then you get to pick which of the protocols. This is our next-gen sequencing workstation, so what we call NGSB. Which of the protocols do you want to be running? on the system, and it will automatically bring it up, and all you have to do is push run. So, this makes it a lot simpler. You have pre-written protocols in the background, and the user interface is a lot more approachable this way. Another example of that would be our assay map offering, which here you can also see the user interface. It's based on the application, so depending on which application, you can go to the app library, select which application you want to run, and once you pick that, again, you have a very nice user interface. It tells you what should be on the Bravo deck and which position, and then you have flexibility of what volumes you want to be using, and it makes the interface much simpler, much more approachable. Next, I have a few slides just because Assay Map is so unique. I just wanted to kind of quickly introduce it. But again, it will depend on your application, so that might not apply to everyone on the call. Basically, it's a chromatography column on the microscale. You can see that we have these cartridges. So instead of a regular Bravo head, we have a unique Assay Map head that is only offered through Agyant. And each cartridge then will be for unique application. It offers, same great reproducibility as you get with Bravo. You have the simple user interface that I've already mentioned, and you can increase your throughput by using this automated solution. Here's just an image if you are wondering how big the cartridges are, some pictures, because that always helps people to understand. At the end of the webinar, you will get a link to our Assay Map page, and there are very nice animations that show you how the cartridges work, and you can learn more about Assay Map if you're interested. I did not want to go through it here because, again, it will depend on the audience and who's doing what. Quick list of the applications that we have. We continue to develop new applications and new cartridges for Assay Map, but here's a list of what is currently available. Again, won't go through each one of them in detail, but just wanted to quickly show this. So whether you're using Assay Map Bravo or just a Bravo by itself, you will get great reproducibility with the system. You will increase your walkaway time, so you're able to do more. If you want to increase it even further, you can, again, build out a workstation that we can work with you in terms of what are your needs and find a solution for that workstation for you. And then we have the software where you can have the flexibility of the VWorks in the background, but what we call the forms is our user interface that makes it very approachable, very easy to use, and also minimizes the training required. I talked about references, so I just wanted to show a few customer references that we have. We have a number of Bravo customer success stories that we have published. And so, again, depending on different applications, what you're doing, you can find customers that are using it. Bravo has really been very well recognized in the industry, used in many, many labs, and whether it's by itself, as an NGS for the next-gen sequencing, or as the Assay Map solution. You can find, I won't go through each one of them and read them, but I just wanted to share it with you guys that there's a lot of happy customers out there. And there's some case studies that will show you then, how much time will you save when you're doing, in this case, Assay Map versus hand pipetting. So again, when you're thinking about automating your liquid handling, make sure you understand what is the end goal here. there will be different solutions that you will need, and you should evaluate whether you're trying to increase your throughput or is it just a better reproducibility that you're trying to achieve. And obviously that will depend on how many samples are you trying to run. The applications, think there was something that's driving your decision today, but also try to think about what you might need it for in the future so that you understand how flexible you need your instrument to be. And again, from hardware perspective, from software perspective. And I think that the ease of use is really a key here. And we see it not just in liquid handling, but in any laboratory instrumentation where you want to make sure that you don't invest in something that nobody will use later. It's got to be approachable. You have to be able to train your staff. People change. People come and go. You've got to be able to train new people and really convince them to use it. Otherwise, you train someone today, and maybe a year from now, or a few months even, they leave. You want to make sure that others can pick it up quickly and put it to use. And then again, I'd encourage you to just look for references. So for questions, as Trevor mentioned, please feel free to enter them into the chat box, and I think we'll do the Q&A at the end of the session. So I will be here, and I'll be happy to answer any questions at the end. Thank you.
Great. Thank you very much, Kasia, and thank you for sending in your questions. Our next speaker today joins us from Biotech. Peter Brescia is an application scientist at Biotech Instruments. He holds a Master's of Science degree from the University of Vermont in Cellular and Molecular Biology and an MBA in Performance Measurements from Champlain College. Peter's current work involves automation of microplate-based assays in the areas of cell imaging and stem cell biology. Thanks for joining us today, Pete.
Thank you, Trevor, and thank everyone for tuning in today. Advance the slide here. Okay, today I'd like to introduce and discuss some of the features of one of Biotech's instruments, the MultiFlow FX, and this is a multi-mode dispenser. And for those folks who have not had the opportunity to see or work with a multi-mode dispenser, I'll start with a short technical presentation, and then I'll move on to discuss some common applications that it might be used for, as well as a few of the available configurations, and then I'll present a few specific application workflows that can take advantage of using an automated liquid handling system such as this. So a multi-mode dispenser, as the name suggests, offers several different dispensing types in a single instrument. And the MultiFlow FX is offered in several configurations, which may include optional modules as well that allow for washing or random well dispensing, RAD technology. And this instrument has become a workhorse in our application labs, where we have several users that come in, and its flexibility allows us to perform a lot of different types of dispensing, and the small footprint allows us to keep the instrument in a laminar flow hood for sterile workflow processes. And the two optional modules that I mentioned, the washer module and random access dispenser, or I'll just refer to it as RAD module moving forward, they extend the capabilities of the base instrument. And the wash module is particularly useful for common processes such as media aspiration or washing steps such as with an Elisa. And they can be performed on a whole or a partial plate, and they can also, these modules can be used in combination with the other dispensing steps. The RAD technology allows delivery of reagent to an individual well as or which I should say may be required when working with assays such as those involving hit picking. So having access to both the peristaltic and syringe pumps for delivery of reagents improves flexibility and helps to optimize assay design. And we'll typically use a combination of those for our applications development. Typically when we're working with precious reagents, the peri pumps are used, and these allow for low priming volumes as well as the ability to run them in reverse and recover any unused reagents for future experiments. And the cassettes that are used with those peri pumps are autoclavable, so you can sterilize the fluid path, and the syringe pumps require a little bit larger priming volumes, so those are best suited for bulk reagent dispensing such as buffers or bulk substrate. addition. And all the fluid paths have angled dispense and washing tubes. And so those are helpful to optimize cell retention when you're performing cell-based assays, which I'll talk about in a bit more detail. So here's a configuration with the instrument. It's offered in a number of different configurations. Here it's shown with the peristaltic pumps, which can be seen on the left-hand side of the image, and the syringe pumps, as well as the optional multi-channel wash module. And here the configuration's been changed slightly to include the RAD module, and users can switch between the options as needed within a particular assay workflow if necessary. And I'll detail a few applications here where these will help, where using a multi-channel dispenser will help improve workflow as well as improve data quality and allow conservation of reagent. So in either configuration described on the previous slides, we have multiple dispensing options, and these are used in a wide range of applications, including primary and secondary screening assays, compound storage methods, genomics and proteomic research, as well as greatly improving ELISA workflows, and we've used it quite a bit here for cell-based washing, fixing, and staining protocols. The RAD module is especially helpful when you need to dispense to random wells, and that's very helpful for saving cost savings with precious reagents and other costly reagents, including antibodies and different types of small molecules. The RAD can also alleviate bottlenecks and workflows that are associated with cell growth assays by automating the cell seeding and media dispensing, or working with various cell culture insert plate workflows where positioning is critical for your dispensing. And also that same technology is helpful when dispensing to non-standard labware, which makes it easy to optimize dispensing there as well. So when two or more dispensing operations are required, the dual peripump configuration allows for quick changes between reagents or to have multiple reagents ready to go. There's two of them available. Again, the ability to autoclave the cassettes resulting in a sterile fluid path is used quite often here in the application lab for cell seeding operations or for media exchanges and adding reagents or compounds that may be in media and require long incubation times. So one couple of applications that I'd like to discuss here. Some recent work that's been performed in the application lab here at Biotech. The multiflow was used for assay optimization and performance of a homogeneous proximity assay, and that was used for the detection of phosphorylated signaling molecules. So cell signaling is typically initiated by cell surface receptor ligand binding and then activation of kinases to propagate a signal via protein phosphorylation in many cases. And this is the case with both the receptor tyrosine kinase receptor, RTKs, and G protein coupled receptors, GPCRs, and their regulation of ERK1/2. Also, and that's shown over here in the left of the screen, upper left. And then TGF-beta signaling through the SMAD family of transcriptional activators is shown on the right. And the reason why we're looking at these two pathways are of interest because ERK1/2 has been shown to play a role in SMAD signaling as well. So we were investigating assay performance in a high throughput 384 well format across several of our instruments, microplate instruments, and we wanted to use automated liquid handling to increase precision. And as these assays had not been performed in a HTS format previously by us, we performed several optimization steps prior to performing any pharmacological studies. Now, both of these assays rely on alpha ELISA technology, and for those who aren't familiar with that, you utilize a pair of antibodies to the analyte of interest, and you create a sandwich-type immunoassay, and that brings the acceptor and donor beads in close proximity. And then once you excite the donor bead, singlet oxygen triggers a cascade of energy transfer in the acceptor bead, which emits light then at 650 nanometers, which would be our readout. So in performing these types of assay optimization steps, typically we'll have to optimize sample and reagent volumes as well as reader settings for assay detection. And here's where the multi-flow can really help reproducibility as well as reagent savings. So typically only a few replicates are used for optimization or a partial plate. And you can use a single or several tubes from a multi-flow cassette, which has 8 channels. And that's typically sufficient to perform the optimization experiments. And then delivery from those can be just several columns across 1 or 2 rows. And in this case, we were performing a standard curve. Each concentration was run in quadruplicate using 2 rows of a 384 well microplate, which is accessed by a single channel of the 8-channel cassette. And the assay itself had 2 dispensers of 5 microliters each of donor and acceptor bead. And we were able to modify the tube lengths a little bit to get our priming volume down to 125 microliters. And much of that can be recovered after for use in later experiments. And so here we can see control lysate standard curves for both assays, and we have excellent signal background covering several decades with excellent correlation between the replicates. So then following optimization, reader optimization and assay optimization, we'll typically run our pharmacology here. We show agonist and inhibitor dose response titrations. These were performed much in the same manner as described on the previous slide. However, being a cell-based assay, once we moved on past the controls, cells were seeded in a 96-well plate format using the multi-flow. And then following drug treatment and some media exchanges, the cells are lysed and a portion of the lysate is transferred and analyzed in a 384 well format. And here again, we'll use our multi-flow for dispensing our 5 microliters of each donor and acceptor bead to conserve reagent. And typically, additionally, we'll run Z prime factor experiments, 48 replicates in a 384 well plate, and positive and negatives controls. And here's where we really see delivery from our cassette, where we can save on priming volume, and we get excellent replication, especially when we're doing low volumes and high density plates. When 2 or more dispensing operations are required for an assay, and 1 or more of the steps includes a bulk reagent, then we'll use our peristaltic pumps in combination with our 2 syringe pumps, and this allows for greater flexibility. And with the addition of the optional wash module with dispense and aspirate capabilities, it really sets us up nicely to run ELISA-type assays. And here is a diagram of the configuration with both peri-syringe pumps as well as the multi-channel wash head. So the wash module allows dispense and aspiration in a variety of plate formats from 6 to 384 well plates, and the volume ranges from 20 microliters to 3 milliliters, so it should cover most applications. And the manifolds, again, are equipped with angled dispense tubes so that we can prevent any disruption of our cell monolayer during washing. So here's an example of an application where this configuration would be useful. In this application, we were comparing 2 relevant assay formats, one being the alpha ELISA format similar to what I just described, and the second a typical sandwich ELISA. And this was for the detection of residual protein A that may be a contaminant of recombinant human monoclonal antibodies prepared for therapeutic use. So as can be seen in these 2 diagrams of the different assay technologies, the alpha ELISA is a homogeneous assay. It requires several reagent additions, whereas the ELISA requires several addition steps but also multiple wash cycles, I believe, 9 in total for this assay. Both of these assays, again, were performed in 384 well format for HTS compatibility, And both had precious reagents, so we wanted to conserve the antibodies and alpha beads that we were using. So peristaltic pumps were used with their low priming volumes. Bulk reagents such as streptavidin HRP conjugate or substrate could be added using the syringe pumps, and all of the wash steps were performed using optional wash module. The use of the peristaltic pumps and cassettes, again, becomes more important when optimizing the assays. As I mentioned, here the reader detection parameters were optimized using a partial plate. Again, now we're talking about coated plates, and this was actually a developmental type assay with one of our partners, and we only had 2 plates to work with, so this becomes critical to be able to use a partial plate. And so we were able to benefit from the using the multi-flow FX and cassettes. And we've seen this time and again where we have improvements between correlation in our replicates when we use automated dispensing versus manual methods. And that was certainly the case here, where both the alpha ELISA and ELISA assays showed excellent correlation between the replicates with only slight variability in replicate data at the lower end of our range that we were looking at. And this results in, of course, lower limits of detection. And based on the standard curve here, we can see we were down in the low picogram per mil range for both assays. Then we went on to screen a panel of 10 antibodies for residual protein A using both methods for comparison, and again across several different readers. You'll see in the table here we were looking at 3 different readers in the 2 different assay formats. All antibodies were sampled in quadruplicate, and residual PA was detected and quantified by interpolation from the standard curves that we ran previously. And then the concentration of protein A is presented in the bar graph as a percent contamination, and you can see we were able to detect very low percent contamination of residual protein A. I'd like to take the last couple of minutes and give you a quick overview of the RAD module and then mention a few applications where this would be most beneficial. There are 2 cassette configurations, a single tube cassette and an 8-to-1 cassette. The single tube cassette has 1 tube and 1 tip for random individual well dispensing, and it has the benefits that I mentioned previously with low prime volume as well as reagent recovery. The 8-to-1 cassette is a little different. It combines all 8 tubing lines to a single dispense head, and this is very useful for rapid dispensing of high volumes into lower density plates such as 6 and 24 well. And again, this can be mixed and matched with the other types of dispensers as well. Either module allows low prime volumes and the ability to access a single reagent vessel as shown in this image here, either the 8-into-1 cassette shown on the left or the dispense channel on the right. And all the dispense manifolds do have the angled tubes, and the flow rate, Z height, carrier speed are all adjustable, and this is very helpful to minimize or to optimize your settings to minimize cell disruption when performing cell-based assays. Here on the right, you can see on the top 2 images, the cell monolayer remains intact, undisturbed when using angled dispense versus the bottom images where we see disruption when using straight tubes. And the rapid high-volume dispensing in a low-density plates is achieved using this unique tip chute. And the tip will direct the flow to the side of the well, and this allows large volumes to be dispensed gently into lower density plates. Last, I'd like to just point out that control of the instrument can be achieved via an integrated touchscreen, and this allows controls of not only the basic functions which I've mentioned, such as priming and purging, as well as programming And then modification of more complex protocols is also available from that touchscreen menu. And there's a flash drive port that's available as well for program storage and then transfer between Biotech's liquid handling control software. And that provides an alternative control mechanism which you can directly control the instrument, as well as the ability to build liquid handling protocols offline. Well, I'd like to thank you all for your time today, and please stay connected with Biotech. Feel free to contact us here at Biotech if you have any additional questions after the conclusion of the webinar.
Well, thank you very much, Pete. And just a reminder that this webinar video will be available next week on Lab Manager's website. And in addition, all of our presenters have kindly provided us with additional resources. They are available through the resource window or the file folder icon, which is located at the bottom of the console. Our third speaker today joins us from Labcyte. Elisa Morales is the Senior Product Manager at Labcyte. She has worked in the life science industry at companies including GE Healthcare, Agyant, and Applied Biosystems. Elisa has a bachelor's degree in molecular biology and a master's in genetics from Smith College. Thank you for joining us today, Elisa.
Hi, good morning, everyone. Today I'll be talking about integrating acoustic technology into your laboratory. When looking at integrating and automation into your laboratory, a lot of labs have goals such as maximizing lab budget, increasing data quality. A lot of labs want to integrate assays quickly into their lab flows, and they want to automate complex assays. In this presentation, I'll show you customer data and customer papers where they've utilized the ECHO to accomplish these goals. We have three products in the ECHO liquid handler product line, the 555, the 550, and the 525. All three utilize acoustic energy to transfer liquids. This transfer has no contact, which helps with potential contamination control, and we do it at high speeds. We use something called dynamic fluid analysis, where we assess the composition of the fluid, which helps determine the power that's needed to eject the fluid into a sample assay plate. We are also able to integrate into our own access automation, which I'll talk about at the end of the presentation, as well as third-party automation. So as a quick overview, I'm not going to go through all of this information. If anyone is interested in the slide deck afterwards, you can please email me. So the 555 is our fastest ECHO. It's typically used in the pharmaceutical environment for high throughput screening. We're well known for that within the industry. This instrument, however, can also be used for cell-based assays, protein crystallography, arraying, and also really high throughput genomics. It is a 2.5 nanoliter drop, which we have high precision, and is very useful when you're trying to save on compounds. With 550, which is a mid throughput speed, that is very, very popular amongst smaller pharmaceuticals and genomics facilities. And it has a broad range of applications such as epigenetics, SNP, utilizing or moving assays for SNPs, etc. And the 525, which is our newest instrument, we transfer larger volumes, 25 nanoliter drops, and it's very, very popular within the genomics space. It's able to let genomics facilities minimize their assays and save quite a bit of money. So how does the acoustic technology work? This is very different than tip-based technology. We use a transducer to generate power, and we can, again, transfer 2.5 or 25 nanoliter drops depending on the instrument. We eject this into the destination plate, and one very popular question is, how does the liquid stay in? The liquid is basically just with tension, and we also have technology that helps the liquid adhere once it gets into the destination plate. And this instrument, our instrument repeats this action 200 to 500 drops per second, which is where we get the very, very high speeds, and we're able to quickly create assays from labs. As I mentioned, we are really well known for high throughput screening. However, this instrument can support a broad range of applications. Again, genomics space, synthetic biology it's becoming very, very popular because of our ability to miniaturize and our very high precision. Proteomics, again, cell-based assays, high content screening, we are able to transfer cells. And drug discovery, again, not just high throughput screening, but compound management and also ADME tox and combination screening, which I'll talk about again at the end of the presentation. Fluids transferred, a lot of people ask because again, when you bring an echo into the lab, we do all the calibrating here in the manufacturing facility, but our instrument can actually do more than DMSO, which again we're known for. We can transfer buffers. We can transfer glycerol up to 50%. We can transfer surfactants, alcohols, and nucleic acids. And the bottom of this slide, you actually see an echo in action. This is what it looks like with a slow motion camera from the time that we create the energy and when the drop leaves. And then again, we do that 200 to 500 drops per second, so very, very high speed. So why would you want to use acoustics versus a traditional tip-based system? Again, better data. We do not have tips, so you don't have to worry about losing compounds and samples due to adherence to the tips. We don't leach anything, so again, there's no contact, no cross-contamination. For increased productivity, again, we can do high-speed assays. We move any well to any well. So that means that you can put all of your different reagents into one source plate and transfer very quickly into the destination plate to create multiple assay conditions to rapidly test downstream to see what works best for your lab. And cost savings, we transfer these nanoliter volumes very, very accurately and precisely, which enables labs to miniaturize their assays without losing any quality of their downstream data. This is a study done by the University of Dundee looking at the cost savings from a traditional tip-based system to the Echo liquid handler. They found two things when they did the study. The first thing they found is that they could create their compound plates very rapidly, 25 minutes on the Echo versus 40 minutes with the traditional tip-based liquid handler. Then they started looking at the cost savings. And what they found is that they would be able to extend the life of their current compound library by 3 to 4 years, thus helping them save over 500,000 pounds in the next 4 years, which probably about $750,000 in U.S. dollars with exchange rates right now. They found that the Echo liquid handler had a huge impact on how they were able to manage their compounds and all of their drug discovery activities. I mentioned precision a lot, but here's actually data from a study looking at the Echo versus multiple nozzle and tip-based devices. And what we found is that the Echo does consistently drop from the 2.5 up to 10,000 nanoliters, whereas the other instruments, once you get into the sub-nanoliter range, begins to have precision and accuracy issues. So again, this precision accuracy will help a lab miniaturize without losing any data quality. I mentioned early in the presentation about loss of sample and compound. Here is a study done looking at hydrophobic compounds on tip-based systems versus acoustic systems. And because there is no contact, you find that there is no loss of the compound, and here you see it as fluorescent signal when the Echo transfers it. However, when the same hydrophobic compound is transferred with tip-based system, you have adherence, and therefore you have decrease in fluorescence. So again, this will impact your downstream studies if you're not getting your sample into your assay plates. This is actually a study we did in-house looking at HTRF assays. And we were looking at data quality when we have a sample done with traditional tip-based versus on the acoustic liquid handler on our echo, and also what happens when you miniaturize the assay. And we found something very interesting. The data quality is comparable, and in one case it's actually better. You actually show higher potency in the drug. And the hypothesis was, because it's a very hydrophobic compound, that again that the compound was being lost in the tip. So as a secondary test, we looked at the same transfer but at 20 microliter volume with the echo and the traditional tip-based, and we again found that the echo showed higher potency. Therefore, we believe that, again, the compound was sticking to the tips, and that will affect, of course, your downstream processes, particularly in drug discovery. Following up on that, MedImmune released a paper that is currently available online on JALA and will be in a special edition of Acoustic Liquid Handling in February. They studied the effect of acoustic dispensing in their entire drug discovery process when they're looking at peptide compounds. And what they found is consistently there was no loss of peptides during the process when using acoustic transfer. They did, however, find peptide loss when they used the tips. And they also looked at not only just tips just in the assay, but they also looked at using BSA and DMSO. With the BSA, they found that the BSA did help when using the tips. However, it did not prevent all nonspecific bindings. There was some loss in potency data. DMSO did perform well. However, if you use too much DMSO, it can negatively affect your assay. But they found that the echo consistently showed no loss and very high potency. And at the data at the bottom, you'll see on the left side, it is the DMSO with assay and the tip-based. You see some loss of potency with the tip-based. On the right, you see the work done on the echo, and the DMSO study and the assay buffer without the DMSO showed exactly the same comparable data on the echo. Therefore, they believe there's no loss of the compound on the tips. And again, that's available on the JALA website. So what does that mean in the greater picture? Here's two reports done by two different pharmaceuticals. The first one's Bristol Myers Squibb. They found 46 additional compounds by using the echo that they would not have found using tip-based methods. And that can have very large impacts, of course, on your drug discovery process. AstraZeneca filed a patent on an entire new set of compounds that were only found by using an echo liquid handler. Again, these were lost during the process when using tips, and again, they were able to find it with the echo liquid handler and create patents for this particular set of therapeutics. So again, tip loss and things like that when you're looking at your stream can really, really impact your downstream research. So integrating is not just looking at the system itself, but also the software. We have several software packages that go along with the ecosystem. They're all created for specific tasks. For example, we have a cherry pick that'll help a lab that wants to create plates that have unique stamps, or maybe you want to pick samples from different source plates and pull them into one assay plate. The software rapidly helps you do that. We also have plate reformat, which is very, very useful in the genomic space. It will help you rapidly stamp out plates or also create unique assay arrangements. Also, all this software is designed to help you really rapidly assemble assays, miniaturize, and move these assays quickly into your laboratory. And we're launching this year something called Echo Combination Screen. It's a new software, and this is really helping particularly any type of lab that wants to do multi-layer assay arrangements. It does all the calculations for you. You basically just lay out the plate and you tell it how you want to arrange your assay and all the reagents, and it will do it quickly for you. This will save you a lot of time with calculations, and it will also give you basically let you do research that you've not been able to do before, typically, this rapidly. And again, it's really focused on combination type screening or labs that want to do highly complex assay development in their lab. And I also mentioned automation. We do have the Access robotic workstation. It is a modular system that accompanies the Echo to help the Echo move basically faster. We have software that's used to really figure out the timings so you have the most efficiency in your lab. We do a lot of customized work. You can add bulk fillers, plate washers, readers, incubators, centrifuges, etc. So again, to help integrate the Echo into your lab very easily and not have to change your SOPs that you're currently using. And thank you. And again, if you would like the presentation, please email me and I'd be happy to send it to you. Thank you very much.
Great, thank you, Elisa. And our final speaker today from Douglas Scientific is Dr. Nico Tsatsos. Nico is an application scientist at Douglas Scientific, where he supports the sales team and clients in molecular diagnostics and genotyping services. Nico received his PhD from the University of Minnesota in biochemistry and molecular biology. His research included gene expression and signaling related to lipid metabolism in mammals. Previously, he worked in the IVD industry, developing new clinical assays for Diasorin and Beckman-Coulter. And Nico also served as an adjunct professor at the University of St. Thomas in molecular biology during his graduate studies. In his recent role, he works closely between chemistry providers and their customers to help meet the application needs of various industries using Douglas Scientific's automated solutions. Welcome, Nico.
Thank you for the kind introduction, Trevor. Hello, and thanks for joining us at today's webinar. Today's labs experience pressure to do more with less, a lot more. Fortunately, technology has evolved to the point where it can adequately and efficiently enable labs to do more, better, faster, and cheaper. But with so many options available, labs also need better information on how to choose a system that accomplishes their goals. Our experience shows that labs need a solution that provides these three key benefits: improved efficiency, fewer errors, and reduced variability. At Douglas Scientific, our mission is dedicated to making the world a better place by enabling the advancement of research at unprecedented speed and fractional costs. Many of you may know us for the array tape platform. We sold our first array tape-based automated liquid handling system in 2007. We were commercialized in 2009 and incorporated in 2010. We are a rapidly growing company with over 105 employees and 175 plus instruments in 15 countries. First, I wanted to describe what array tape is for those who may not know. Array tape is a thin and flexible microplate replacement and could, for example, be used in place of a PCR plate. Array tape comes in the form of a continuous polymer strip, serially embossed, and reaction wells in 384 or 768 well formats. There are anywhere from 50 to 200 arrays, or microplate equivalents, spooled on a single compact reel. An array tape is customizable in terms of well shapes, volumes, and formats. The most common solution is our polypropylene 384 well array with a 2 microliter well capacity and a 1.6 microliter combined reaction volume, which results in significant savings for labs that are currently working with much higher volumes, 30, 20, or 10, even 5 microliter reactions. The array tape platform is comprised of the lab automation solutions for labs with medium to what we've termed ultra-high throughput requirements. Significant return on investment can be seen from 100,000 data points per year to over 40 million plus. The array tape platform is chemistry agnostic and very flexible to set up only the reactions you need. On the far left of this figure, the IntelliCube by itself utilizes a Peltier block inside the IntelliCube and is used for either qPCR or endpoint PCR reactions. On the far excuse me, in the middle, the IntelliCube with our IntelliCycler water bath increases throughput for endpoint PCR workflows. And on the far right, the Nexar also utilizes an offline water bath thermal cycler in addition to a fluorescence reader, allowing for maximum throughput using array tape. Both systems do have small tip consumption, but tips typically only need to be changed weekly by utilizing the onboard pipette wash feature for both instruments. The IntelliCube and Nexar support PCR-based applications while improving the efficiency, minimizing the errors, and reducing variability. So let's talk about how efficiency comes into play. The inline automation of the array tape platform enables you to increase throughput by 2 to 10-fold due to its inline automation. A lab can save up to 90% on reagents. The most common reaction volume, as I stated, is 1.6 microliters. Typically, we'll see a 800 nanoliter sample dispense followed by an 800 nanoliter reagent or PCR master mix dispense, which typically is a 10 to 20% usage relative to standard microplate PCR setups. This is also important for people with precious samples or controls, as they can conserve those samples using these lower volumes. In addition, offsetting labor costs by 60% or more due to streamlining our process. You eliminate the bottlenecks and make the most efficient use of your lab personnel. The array tape platform significantly reduces the need for manual plate handling and even some of the other robotics. As an example, this table that I pop up here shows how one of our customers was able to go from less than 1 million to over 50 million data points using our system with 12 fewer full-time employees and with an estimated reagent savings of 80%. We all know that very few scientists went to school so they could pipette for the rest of their lives, so we want to make a difference in your lab, and the best way to enable that is to free up your time so you can focus on other important tasks in the lab. In PCR workflows, labs can gain efficiencies by automating the sample as well as master mix or reagent pipetting. With sub-microliter volumes, an automated piece of equipment is more efficient than human hands, and further efficiencies are gained by incorporating more automation components, such as automated tip changes. We have 384 or 96, automated sealers, plate stackers or hotels, thermal cyclers, and detectors, and all of these components are most efficient if incorporated into the same system. So let's take a quick look then at what the common lab workflow would look like with these automation components. The top half of this slide displays a common workflow that we frequently see in other labs and that people in the audience may recognize. Both parts of this figure contain the DNA extraction assay setup and still consist of formulating a sample plate and assay plate that you would then put into a downstream process. In the top half, typically people are using liquid handlers to set up their PCR plates and then load those PCR plates either with a plate shuffler or manually onto PCR instruments, as shown on the far right of the top half there. Underneath this common workflow, we show the IntelliCube workflow. And if we compare it to that above, you'll notice that the liquid handling, the plate sealing and shuffling, as well as thermal cycling, represented by the bold blue arrow up top, are now replaced with one fully integrated piece of equipment. This is what makes the IntelliCube unique and essentially removes what we like to call islands of automation using one instrument. The throughput hours, shown on the far right of the slide in green, were calculated based on labs running PCR for a 60 SNP or single nucleotide polymorphism, basically 60 unique reactions that they're running per sample workflow using the 384 well thermal cyclers on top compared to the IntelliCube using the unique 768 well array tape on the bottom. So with a more automated workflow in place, your labs will begin to recognize a reduction in errors. In PCR workflows, labs rely on liquid handlers to reduce error by providing automated barcoded tracking of plates and pipetting steps, eliminating errors introduced by the human hand, such as the wrong volume setting or inaccurate dispensing, sample and assay tracking and logging of files for verification, as well as validated methods That is, once your method is established and validated with an automated system, it is consistently duplicated and repeatable as opposed to manual setups. This not only ensures reproducible workflows, but also allows for easier training of any new personnel. So growing labs are always trying to determine whether liquid handling or a combination of liquid handling and ancillary automation will address the sources of error they are trying to reduce. Some examples we often hear from customers that they desire to be automated to reduce error are the plate tube labeling process and traceability, assay and sample dispensing, generation of plate maps, sealing of reaction plates, plate handling, which involves loading of plates onto the instrument, removing operator errors like dropping of plates, import of plate layout, defining proper plate thermal cycling conditions, etc. Ideally, all these steps are integrated. Lastly, labs are always looking to reduce variability in all their procedures so that they ultimately get reduced variability in results. Again, in the PCR workflow, labs rely reduce variability using liquid handlers results from consistent volume delivery, especially when low volumes are needed. Removing variability from different lab technicians using dissimilar techniques. We all know that even with well-established SOPs, person-to-person variability will still exceed that of automated options. And lastly, ensuring that plates are set up consistently. Plate maps and pipetting patterns are predetermined by existing methods, removing any variability that may be present with multiple operators. Lab managers most certainly want to fully understand where operational variability is most prevalent. Perhaps the intended volume is below what can reliably be manually pipetted. If so, what automation is needed to meet this need? Will multiple operators introduce further variability to the process? More specifically, will liquid handlers effectively address reduction of variability? Is contact or non-contact dispense technology needed? Or consider viscosity. Labs requiring application flexibility would be well advised to select a liquid handler capable of working reproducibly across a range of liquid types. Now that I've covered the three areas liquid handling improves workflow in the lab, I'd like to stress the unique features of the IntelliCube and show you how the PCR workflow is handled on this automated instrument. By integrating liquid handling, amplification, detection, and data analysis all into one instrument, the IntelliCube is the first PCR instrument for laboratories to fully automate this application. Sample extraction plates and PCR assays are loaded onto the IntelliCube, and the machine literally does the rest. I'd like to direct your attention to watch the following video clip demonstrating this integrated workflow.
Introducing IntelliCube. The process begins with independent aspiration of the DNA sample and reagents from the respective source plates. Source plates can be randomly accessed through the plate stacker located on the instrument. The DNA samples are dispensed in up to 384 wells at one time with the Cybe Felix dispense pipette. Following sample dispensing into the array tape, reagents are loaded into the wells. The array is automatically sealed with a pressure-sensitive seal prior to thermal cycling. In the meantime, integrated wash stations are used to clean the tips and provide stringent cleaning measures to mitigate the risk of cross-contamination between samples. A Peltier block facilitates the amplification and supports multiple array formats without block changes. Real-time fluorescence detection is supported with a high-resolution CCD camera and LED excitation source with 5 optic channels and the ability to multiplex data capture in 15 seconds or less. The IntelliCube software supports protocol generation and subsequent data analysis of PCR results. Lab personnel can observe amplification curves on the instrument in real time. Walk-away operation, increased efficiency of lab personnel, increased throughput, reduced chemistry costs, and reduce consumable costs. IntelliCube, your solution for the most efficient lab possible.
Okay, are we back?
We are back.
All right. So in closing, I ask, how does the IntelliCube overcome these challenges presented to you all in your labs today? For efficiency, the inline automation of the array tape platform enables you to increase throughput by 2 to 10-fold. You can save up to 90% on your reagents and offset labor costs by 60% or more. For error reduction, using a fully integrated multi-step PCR workflow, the liquid handling, plate shuffling, sealing, thermal cycling, and data analysis are all in one instrument. Finally, your reduced variability would come from the consistent delivery of samples, consistent delivery of your assays or master mix, and array layouts in either 384 or 768 wells, optimized by our software included on the IntelliCube. So with that, I'd like to thank you for your time today, and I'll hand it back over to Trevor.
Thank you very much, and thank you all for your presentations. I believe we'll go directly into our question and answer session. We do have a number of questions that have come in from the audience. And the first question, perhaps I can direct to you, Kasia. You mentioned walkaway time. What kind of time savings can you expect from using a Bravo system?
Well, it would really depend on your application. I mean, each case is really, depending on the protocol. But on the Bravo, you can, there's a number of accessories that you're able to mount on deck. You could see some of them when I was showing the assay map slides. It's anywhere from filtration. We have a vacuum filtration to mixers to heaters, coolers, and more. So you can really automate quite a bit of your process, and with the protocol, then you'll free up quite a bit of your time as the samples get processed. But again, the specifics will really depend on the exact protocol that you're trying to run.
Okay, thank you. Now, is it difficult to integrate products from different vendors with these systems?
So that will depend. For the accessories that I just mentioned, that we have on deck, so we basically can put anything that has the SLAS footprint. I'm sorry, the SBS footprint of a plate. We can put that on deck. We have different also mounting. We work with other vendors to allow for their tools, I guess, to be part of the the accessories that we offer. If you're trying to integrate with something that's off deck, then you would have increased costs because you're dealing with probably an arm. So you can, like a robotic arm. So you might be able to do it with, like, when the workstation that I showed has the bench cell, or you might need a bigger workstation, in which case it's doable. It's just a matter of cost. But, we found with some of our customers, when we talk about sample prep, whether it be for the LCMS workflow or even if you're trying to normalize some samples, say in front of a plate reader, sometimes, if customers are really focusing on increasing the reproducibility, they don't mind, and they have limited budgets, they don't mind being the robotic arm and moving the plate when it's ready from the Bravo to, say, plate reader, or moving the samples to LCMS. But it's really, about the precision and controlling that day-to-day variability that two of us here spoke about, that you can improve that with automation.
Thank you. Now, Elisa, does the dispensing occur well by well with the Echo system, or can it dispense multiple wells simultaneously?
It's a well by well dispense, but that basically we do it very, very rapidly. So say a 384 well plate with, let's see, say a 384 well plate, about 100 nanoliters will be less than 2-minute cycle time. So that's when it comes in to going out.
Okay, thank you. And what are the transfer ranges for the Echo?
The transfer range for the 555 series is 2.5 nanoliters up to 10 microliters. For the 525, it's 25 nanoliters up to 10 microliters.
Great, thank you. Now, Peter, you mentioned that the fluid paths can be sterilized on the multi-flow. How exactly is that done?
The cassettes themselves are autoclavable, so they are just positioned in the instrument at time of use. And so typically what we'll do is we wrap them in tin foil and we'll place them in our autoclave for 20 minutes. And you can do that multiple times. We recommend up to 50 times per cassette. The bulk dispensers are a little more difficult, typically because they cannot be autoclaved the entire pathway, and the way that we sterilize those is to run an ethanol solution through them. And we've done that successfully for bulk reagent dispensers as well for the syringe drives.
Great, thank you. And now how quickly could you dispense, say, 5 microliters to a 384 well plate?
Using a 1 microliter cassette, it takes approximately 5 or 6 seconds, closer to 6 seconds on a 384 well plate. The cassettes themselves come in 3 different sizes, 1, 5, and 10 microliter cassettes. The 5 microliter cassette, you can dispense 5 microliters in about 6.5 seconds.
Great, thank you. Nico, how difficult is it to optimize reactions for miniaturization?
Yeah, that's a great question. Our scientific team is well equipped to assist our clients with assay optimization, but fortunately we're seeing that very little optimization is required with most assays. Occasionally we'll maybe encounter a particularly finicky assay that needs some tweaking, but this is usually quickly addressed by an adjustment in the primer probe ratio or sometimes in the temperature settings of the PCR profile, and we have a team happy to assist with that.
Great, thank you very much. And for Elisa, one of our viewers asked, can MTBE be used with the Echo system?
I will have to look into that and get back to them. MTBE, write that down. I'm not sure.
All right. Well, we'll forward that question on to you.
That'd be great, and I can ask our chemical scientists. Thanks.
Absolutely. While I do have you, though, what software do you recommend for assay development on the Echo?
Echo plate reformat is typically what we recommend for assay development. It will let you set up, again, multiple conditions within one plate, and you can also put in, say, something with 50% glycerol right next to something with detergent, whatever you need, and then the instrument will be able to go, because it does one well to one well, be able to transfer quickly based on whatever plate design you do. And because of the DFA, we'll be able to adjust well by well depending on what reagent's in there, which again will let you rapidly create that assay.
Great, thank you. Now, one of our viewers is looking for a solution for sample prep of blood for toxicology. Are any of these systems compatible with that application?
Not on my end.
No? Kasia?
I'm sorry, can you repeat the question, please?
One of our listeners is looking for an automated solution for sample prep of blood for toxicology. Is that something that Agyant would have a solution for?
We have worked with various types. I think we've looked at blood, and we've looked at, there's a lot of urine preparation for LCMS testing. So I would, yeah, we might have a solution for you. It will depend on the protocol. Again, depend on your needs in terms of how closely do you need to track each sample and what you need to do with the blood. But we can definitely look into that.
Great, thanks very much. We'll forward that to you, and you can follow up with them. Nico, do the benefits of automating qPCR really outweigh the added costs?
Yeah, certainly. On the surface, lab automation is really thought of as a bargain, but when you factor in the benefits of removing the human error and gains in efficiency, it becomes quite clear. The IntelliCube goes a step further by drastically reducing the use of the reagents, especially for our PCR reactions and qPCR. Those tend to be the very expensive components. And we can frequently get the cost per reaction down to less than 10 cents a determination. And in many cases, this is the huge savings that justifies the investment almost immediately.
Thank you. Now, Elisa, do you have to train the Echo how to transfer new liquids?
No, the Echo is calibrated with wide ranges of liquid classes. And what the machine does with the DFA is it interrogates the fluid, figures out the composition, and then figures out the power. So there is no calibrating within the lab.
Great, thank you very much. And as I look at the clock, I think we're running low on time. We may have time for one more question. Peter, can the multi-flow be integrated into larger automation systems?
We do work with several automation partners, and they certainly have been integrated. They do have the standard microplate footprint that Kasia mentioned. So yes, those as well as our washers and readers have been integrated into several systems, larger systems.
Great, thank you. And thank you all for the discussion today. This brings us to the end of today's webinar. If you have any further questions, please reach out to our panelists directly. Their contact information, as you can see, is on the screen now. And again, just a reminder that today's webinar video will be available at the link you see on screen now. On behalf of Lab Manager, I'd like to thank our panelists today for the hard work that they put into their presentations. And I'd also like to thank our sponsors, Agyant Technologies, Biotech Instruments, Labcyte, and Douglas Scientific for supporting our Tech Trends webinar series. As well, I'd like to thank all of you for taking time out of your busy schedules to join us today. Please mark your calendar for our next Tech Trends webinar, which will focus on clinical chemistry techniques. This webinar will be taking place Thursday, September 24th from noon until 1:30 p.m. Eastern Standard Time. And for more information on this and other webinars, please visit our website at www.labmanager.com. We hope you can join us again. Thank you and have a great day.